Okay, so we just ended our unit on carbohydrates. Now we're moving on to lipids. We talked about the 4 organic molecules that we are going to be covering for the unit on biochemistry, which are carbohydrates, lipids, nucleic acids, and proteins. So now we're going to take off with lipids. So lipids contain carbon, hydrogen, and oxygen atoms, just like carbohydrates, but they have less oxygen proportionately. So all lipids are nonpolar. So what does that mean? So nonpolar molecules do not have a charge on either end of that molecule. They are neutral. Lipids are insoluble in water. And we are going to be covering the 4 categories of lipids: triglycerides, phospholipids, and waxes. Those are fatty acids attached to an alcohol. And that last category is steroids, which are composed of 4 fused carbon rings with different functional groups attached. So if you remember the last functional group we talked about, it was a hydroxyl group, which you should remember was OH. So triglycerides include lipids that we know as fats and oils. They form from a reaction between glycerol. So what kind of molecule is glycerol? It ends in OL, which tells you it will be an alcohol. So triglycerides form from a reaction between glycerol and 3 long-chain fatty acids, hence the prefix tri. So that is your glycerol there. It's a 3-carbon backbone. It has 3 hydroxyl groups and then the surrounding hydrogens. So fatty acids are made up of a long hydrocarbon chain attached to a carboxyl group. As we say groups throughout biochemistry, when you hear that, that's just going to be another functional group. You guys will see what carboxyl group is. As far as hydrocarbon chain, that consists of carbon with hydrogens attached to it and bonded to it. So what is a carboxyl group? It's COOH group. And the hydrocarbon chain is nonpolar and hydrophobic. The carboxyl group is polar and hydrophilic. So let's just stop for a second and talk about those 4 words. So we said nonpolar has no charge on either end of it. Hydrophobic. Well, if you think of hydro meaning water and phobic meaning fearing, it's technically, we call it a water-fearing molecule. It's not soluble in water. The carboxyl group is polar, meaning that that end will have a charge to it. And hydrophilic, hydro meaning water, philic meaning love. So we call that like a water lover. Things that are hydrophilic will be soluble in water. So this is showing you the ends of that fatty acid. So we have right here our carboxyl group. That C is double bonded to the oxygen, and then there is an oxygen and a hydrogen coming off of that. Just like you had built in your carbohydrate building lab, you saw that in your straight chain glucose. That is a hydroxyl group there. This is showing you the separation between the polar part of the molecule and since it is polar, we know that it will be hydrophilic, soluble in water. Nonpolar is this long hydrocarbon chain. Hydrocarbon because it's just carbons linked together with hydrogens attached to them. Hydrophobic, meaning will not be soluble in water.
And why do you think that the polar end is soluble in water and the nonpolar end is not? If you read the section in your book on water and polar molecules, you should know that water is also polar, meaning it has a partial negative charge on the oxygen and a partial positive charge on the hydrogen, and polar substances are attracted to each other because of their charges. Water is polar, the hydrocarbon chain is not polar, so they will not be attracted to each other.
So we have some distinctions between the fatty acids. We have saturated fatty acids. Those have single bonds between all of their carbons. We say that they're saturated with hydrogen. The saturated fatty acids that we think of are the solid fats, more animal fats like bacon. Unsaturated fatty acids have double bonds between at least two of their carbons, so they're not completely saturated with hydrogen. Those are more of our liquid oils, olive oil, vegetable oil, things that come from plants as opposed to animals. Monounsaturated says that there will be one double bond. Polyunsaturated, there are two or more. So that top there is a saturated, right? It is fully saturated with the hydrogens. Below we have our unsaturated fatty acid. And it is a what kind of unsaturated fatty acid would that be? Monounsaturated. There is just one double bond. Polyunsaturated, we would see more than that. So back to dehydration synthesis. You just couldn't get away from it. So it happens when what? When we're removing a water to build a molecule. So it happens when the hydroxyl groups of glycerol. What's a hydroxyl group, which you all are pros on? An OH group. When that combines with the hydroxyl hydrogen of 3 fatty acids. So here's a picture here. We have another video that goes into detail about how this is done. But again, if you picked up the carbohydrate dehydration synthesis, there is nothing more to this. Just knowing that you cannot pull an OH and an H from whichever hydroxyl group you want. The hydroxyl group that you take entirely must come from glycerol. And then, of course, instead of the 1 H2O removed, we have 3. And you can see that new triglyceride molecule, and you can see where those waters were removed. Phospholipids. I think you guys have been hearing about phospholipids since probably 6th grade. They are different from triglycerides because why? They have 2 fatty acids attached to glycerol instead of 3. And in place of that 3rd fatty acid is a phosphate group, hence why it has its name. And they make up the cell membrane. And we will talk more about this later. There is a picture of your phospholipid there. We see the fatty acid tails. This double bond here tells us what? It has 1 unsaturated fatty acid and 1 saturated. And then we see that phosphate group at the top.
And here is the glycerol backbone, although it may be a little hard to recognize because of where the hydrogens are, but you should see that it's 3 carbons in a row surrounded by hydrogens. So you have the 2 fatty acid tails, the glycerol backbone, and then the phosphate group. If this were a triglyceride, that other fatty acid tail would be coming down from that carbon, but it's not. So we have the phosphate group. The next category we're doing is waxes. All you need to know about waxes is really what they look like. They're very kind of complicated structures. They are long fatty acid chains attached to a long alcohol chain. You should also know the functions. They are a protective layer in leaves, earwax. They coat the feathers of birds to make them water repellent. This is what they look like. Just know whenever you see a very, very complicated structure like that, it's going to be a wax, not a phospholipid, not a triglyceride, and not a steroid. You will not need to draw or memorize any of those. The last category we're going to do is steroids. And steroids have a very unique and recognizable 4-carbon ring shape. Some examples are hormones and cholesterol, and I'll show you the shape of it in just a moment. The important steroid cholesterol in living things is produced in your liver, and it has some very, very important functions that you will need to learn. The first one is it does form part of the cell membrane. When we talk about the cell membrane, we're going to talk about the important purpose that that serves. It also is the building block for many other hormones. And lastly, it plays a large role in building nerve cells. So when you see this structure here, you see these 4 rings, 1, 2, 3, 4. When you see those, what are called fused carbon rings, you automatically should recognize this as some sort of steroid molecule. In this case, this one is cholesterol. These are some other examples of different steroids. There's progesterone, let's see, cholic acid, corticosterone, and another cholesterol. So as you can see on this picture, the 4 fused carbon rings are very recognizable, and you shouldn't have any trouble distinguishing those from your triglycerides, phospholipids, or your waxes. They are completely different structures, and when you identify these, you should be able to do that based on the carbon rings. So that is our lecture on lipids. You should now watch the video on the dehydration synthesis and hydrolysis of lipids. Hopefully it will be much easier for you since you are all well versed in the dehydration synthesis and hydrolysis of carbohydrates. It works very much the same way.
Yes.
Ms. Rigoli says yes.